Okay, in this video we are going to be drawing a cell in the parts of the cell, or the organelles. So your supplies you might need, if you just want to draw with a pen or a pencil, that's fine. I'm going to be using some markers and highlighters, whatever I find around my house. So first we're going to start by just drawing the cell and the cell membrane here. So it doesn't have to be perfect, and this is just one cell. So we're going to draw our organelles in here and then kind of label out to the side. So this here is our cell membrane. We also call it our cell wall, but this is creating a boundary between the inside of the cell and the outside of the cell. Next, we're going to draw our nucleus. So it doesn't matter where you draw it within here. I'm just going to draw mine kind of right in the middle here. And then we're going to draw our line and label that guy. So this is, again, our nucleus, or the brain of the cell. N L, there's an L in there, sorry. So N-U-C-L-E-U-S. Sorry about that. Then I'm going to color that in. Doesn't matter what color. I'm going to grab a green. Someone said this kind of looks like an avocado, or maybe a fried egg. So you can think of the nucleus as the pit of your avocado. Then I'm going to color in all of this blank space. You can also leave it blank if you want, but this is called your cytoplasm. It's kind of like the jelly-like substance that fills up the whole cell. So I like to think of this as like Jell-O that's in the fridge that's not completely like firmed up yet. It's kind of just some jelly in there. It gives some structure to the cell and a place for the organelles to kind of flow through. So it doesn't have to, again, it doesn't have to be perfect. Colored in. So I'm going to label that next. And again, it doesn't matter if you want to label it over here or label it up here. I'm just going to keep going down. And this, again, is the cytoplasm. C-Y-T-O-P-L-A-S-M. Now we're going to start drawing some organelles. So first we're going to go with the centroles or centrioles. They look like rods almost, or almost like they're just kind of like straight lines, almost like a 3D licorice type thing. If you really just want to draw some lines, that's fine. They're more like, they're just so you can tell the difference. They're kind of just lines. Doesn't have to be perfect. I'm going to label that guy here. I'm going to go straight up. And the purpose of these is to aid in when the cell splits. This helps with that cell division. Okay. Next, I'm going to go right in here, and that's going to be our Golgi apparatus. So it kind of just looks like layers, so almost like if you draw like half of an evergreen tree. And then I'm going to go back in and just add some, like, a different color to it. So it's folded up layers of like a membrane. And what the Golgi apparatus does is takes either proteins or different different things and substances that the cell makes, takes them in, packages them up, and then sends them throughout the cell. So kind of like the Amazon delivery packaging plant of the cell. So again, Golgi apparatus. G-O-L-G-I-A-P-P-A-R-A-T-U-S. Golgi apparatus. We already drew our cell membrane, so that's a whole big circle around here. Next step, we're going to draw some ribosomes. So these just look like dots all around. So dots. and they kind of like live in little groups. They kind of float throughout the cytoplasm. The purpose of ribosomes is to make proteins. We're going to be adding a couple more in a little bit, but we'll start with that. I'm going to label mine off of here. So I'm going to point at this guy here, and this is a ribosome. Ribosomes. Look at these blue guys there. Again, their purpose is to make proteins. You need to spell that there. After that, we're going to draw a lysosome. So a lysosome, I'm going to throw mine, let's see, right in here. The point of a lysosome, actually, I'm going to bring it down here. It kind of looks like a liver, like a human liver, just kind of like a blob that has a little dent in it. I'm making mine orange and coloring it in. The point of, or its purpose, is to produce enzymes so the cell can break down its nutrients to use as energy. So it produces enzymes for the cell, just kind of like how our liver does. And again, that's called a lysosome. So I'm going to draw my label out this way. So we got L, Y, S, O, lysosome. L-Y-S-O-S-O-M-E, lysosome. Next up is our endoplasmic reticulum. So we're actually going to draw that in a couple places, and it kind of just looks like tubes or lines. We're going to start with it around, and you most often see it around the nucleus. Kind of just looks like a bunch of different lines that connect. But then you also might find it in other places around the cell. It's kind of like our, they're kind of like tunnels that the information and substances travel throughout the cell. You'll kind of see them around. And again, these are all like super microscopic, so you can't see these with your naked eye. You need, this is one individual cell. So kind of just, again, it doesn't have to be perfect. That is your endoplasmic reticulum. There's actually two types. So first I'm going to create a rough endoplasmic reticulum. Rough meaning it has ribosomes on it. So the rough is just calling out the texture that it looks like, or like the dots on it. Then we also have smooth endoplasmic reticulum. So I'm going to leave these lines without the dots on it because this would be smooth or there's no ribosomes on it. So I'm going to actually have two. I'm going to bring one out here to label and then one down here to label. So I'm first going to label this one. They're both endoplasmic reticulum, so I'm going to kind of have to draw through my centrioles here. So I'm going to bring my line out here, and again it's endo. Endoplasmic reticulum. We also might say ER for endoplasmic reticulum. This here, since it has the ribosomes, we're going to call it rough. So rough ER because of the ribosomes. Then we're going to label this one down here. This is also endoplasmic reticulum. but this does not have the ribosomes. So again, it's still called ER for endoplasmic reticulum, but this is smooth because it doesn't have that extra texture. So again, both endoplasmic reticulums, both points are to, or the job of them is to create kind of like a tunnel system or like paths for substances to travel along to get to different places in the cell. So for example, the ribosome might create or make protein. That protein travels through the ER to the Golgi apparatus where it's packaged up and sent off back to the nucleus. It comes through the endoplasmic reticulum into the nucleus. So it's kind of like a gateway or a pathway and door into the nucleus and into different parts of the cell. So protein's important for building blocks of a lot of things, but mainly a lot of protein is used to make DNA and RNA within the nucleus. So you can even make that note there. This is where the DNA and RNA of the cell is, or kind of like its identity. All right, a couple more. We are going to do the mitochondria now, and I'm going to do those like right in here. We have two more. We have mitochondria and a vacuole. So we're going to put mitochondria here and put our vacuole somewhere in here. So mitochondria, I like to think of these as like the powerhouse or the battery of the cell. They are going to make the energy for the cell. So they're kind of rod-shaped, but a little bit thicker and longer than these centrioles. I don't know why I just like make little lines in them. So again, these are called the mitochondria. Mitochondria, powerhouse of the cell. It's going to make energy for us. Right now, this content of this video is just a straight up label. I will make another one that talks more about the actual functions of these guys. Finally, we got the vacuole. So I'm going to put my vacuole right here. Kind of looks like a little kidney-shaped thing too. The point of this, it's actually not filled. It's empty, meaning it has different fluid in it than this cytoplasm. It stores waste. It stores extra fluid in the cell. So I think of the word vacuum, or like it kind of cleans up the cell, or at least holds the waste until it's ready to leave the cell. Vacuole. So like that word vacuum. So it's kind of an empty, hollow organelle. All right, so that's pretty much it for that. So we just labeled all the different parts of the cell. As I'm practicing and learning them and the names, I usually will write or draw this out a couple times, either in a notebook or scratch a piece of paper until it kind of starts sticking in my brain. I'm kind of just literally talking through it as I'm drawing it out. All right, thank you, and let me know if you have any questions. I'll try to back it up for a second so you can see the whole thing, maybe. I'll just take a picture of it.